The global incidence of immune-mediated, metabolic, and neurodegenerative diseases is climbing at a steep, measurable rate. This rising baseline of metabolic morbidity leaves human populations critically vulnerable to secondary physiological shocks and infectious diseases. For instance, clinical data from the original SARS-CoV-2 outbreak shows that diabetic populations faced double the mortality rate of the general population. Connecting these seemingly disparate conditions is a highly active, hidden variable, gut microbiota dysbiosis. Dysbiosis is an ecological state shift within the body, defined by a sharp decrease in microbial diversity and the unchecked aggressive overgrowth of harmful taxa. The microbiome is a multi-kingdom ecosystem comprised of fungi, protists, archaea, and viruses, all residing and interacting within the gastrointestinal tract. To understand how this drives systemic failure, we must map the exact mechanics of dysbiosis, how environmental triggers collapse the barrier, how microbial metabolites dictate host survival, and how we engineer this ecosystem. The gut microbiome serves as the central, modifiable node dictating the severity and progression of nearly all systemic human diseases. A healthy intestinal ecosystem relies on strict spatial separation. A dense mucosal layer functions as a physical shield, safely containing microbes within the gut lumen. The host actively maintains this barrier through specialized Paneth cells, which continuously secrete antimicrobial molecules, including defensins and lysozymes, to prevent bacterial encroachment. While human genetic predisposition remains relatively stable across generations, the microbiome is highly volatile. Profound structural shifts in this ecosystem are rapidly induced by external environmental inputs. High-sugar, low-fiber Western diets serve as the primary macro-level disruptors, reliably triggering intense intestinal inflammation and compromising the mucosal barrier. Damage is also driven by micro-level disruptors. Additives and preservatives, widely assumed to be biologically inert, actively degrade the gut ecosystem. Non-caloric artificial sweeteners actively promote the overgrowth of proteobacteria and induce glucose intolerance, causing the exact metabolic dysfunction they were designed to prevent. Common dietary emulsifiers further compromise the system by directly altering the microbiota composition to favor species that drive local inflammation. This results in severe physical damage, as overrepresented mucolytic bacteria, such as Ruminococcus glavis, aggressively consume and degrade the host's protective mucus layer. Routine dietary choices and highly processed food additives actively engineer the collapse of the host's primary immune barrier. Once the mucus layer degrades, the immediate consequence is increased intestinal permeability. The tight junctions holding the intestinal wall together fail, allowing molecules to leak directly into the host's bloodstream. This collapse causes a rapid depletion of protective outputs, specifically short-chain fatty acids. Short-chain fatty acids, or SCFAs, are organic molecules produced by the bacterial fermentation of dietary fibers in the colon. Concurrently, there is a massive spike in pathological microbial metabolites, which flood through the broken barrier and enter host circulation. These leaked molecules hijack host immunity via toll-like and nod-like receptor signaling. Toll-like and nod-like receptors are critical proteins the host immune system uses to detect microbial invaders, and their overactivation drives systemic inflammation. We can observe the mechanics of this pathogenesis by isolating the choline pathway. It begins in the gut lumen, where commensal microbes enzymatically convert dietary choline into trimethylamine. Trimethylamine, or TMA, is a toxic byproduct of bacterial metabolism. This TMA enters host circulation and travels directly to the liver, where it is oxidized into TMAO. The clinical endpoint of this cascade is severe. High levels of circulating TMAO are heavily implicated in advanced atherosclerosis, while the raw TMA drives direct nephrotoxicity and cardiotoxicity. A compromised gut barrier facilitates the mass entry of rogue metabolic signals into the bloodstream, driving systemic multi-organ failure. Diagnosing the severity of complex metabolic diseases traditionally requires invasive physical tissue biopsies from the affected organs. Because the microbiome constantly sheds specific metabolic outputs into host circulation, we can read these circulating molecules as highly precise, non-invasive biomarkers. This diagnostic methodology is currently being validated in conditions like NAFLD. Non-alcoholic fatty liver disease is a condition characterized by excessive fat buildup in the liver of people who consume little to no alcohol. This chart shows a highly specific 37-strain metagenomic signature napped directly from the gut. By reading this signature, clinicians can accurately detect advanced liver fibrosis without performing a physical biopsy. Within that signature, specific taxonomic indicators are tracked, such as a dangerous rise in valenality occurring alongside a sharp drop in anti-inflammatory fecal bacterium prausnitzii. This graph shows the secondary diagnostic layer, the circulating microbial metabolites, including severe spikes in compounds like succinate and phenylacetic acid. Extracting and verifying these complex multi-omic signatures requires advanced laboratory phenomics and deep sequencing of patient samples. By decoding taxonomic and metabolic signatures in the blood, clinicians can mathematically reverse engineer the biological state of a failing internal organ. Once dysbiosis is accurately mapped, the clinical focus shifts from precise observation to active, targeted reprogramming. The most aggressive intervention is whole ecosystem replacement, primarily achieved through FMT. Fecal microbiota transplantation is a procedure that transfers healthy donor stool into a patient to restore bacterial diversity, yielding a greater than 90% cure rate for severe recurrent C. diff infections. While ecosystem replacement treats catastrophic local infections, complex metabolic diseases require precision metabolic reprogramming. A prime example of this is the molecular targeting achieved by DMB. 3,3-dimethyl-1-butanol is a synthetic compound structurally similar to choline that selectively inhibits bacterial enzymes. Acting as a structural analog to dietary choline, DMB non-lethally jams the bacterial enzyme, completely halting the production of toxic TMA at its microbial source. If bacterial pathways cannot be safely blocked, intervention moves to a third tier, host receptor modulation. We bypass the microbes entirely and upgrade the host's chemical response. This is demonstrated by obetilyl acid, a molecule that potently activates the farnesoid X receptor in the human liver. The farnesoid X receptor, or FXR, is a nuclear receptor in liver cells that regulates the synthesis and transport of bile acids. By chemically activating this receptor, the drug regulates bile acid synthesis, directly ameliorating hepatic steatosis and reversing accumulated tissue fibrosis. Microbiome therapy is rapidly evolving from the blunt application of basic probiotics into the targeted pharmacological interception of highly specific microbial pathways. Dysbiosis functions as a compounding, destructive feedback loop between the gut and host organs. As the liver becomes diseased and steatotic, it loses its mechanical ability to send regulatory bactericidal fluids, like bile acids, down into the gut ecosystem. The immediate consequence of this missing chemical regulation is severe ecological collapse, allowing rogue oral microbes, such as Streptococcus, to colonize the lower gastrointestinal tract. These newly established colonies then synthesize and pump an accelerated volume of toxic metabolites back up to the already failing liver, completing the toxic loop. Pharmacological strategies must treat the gut microbiome as the primary target for intercepting systemic disease. Mastering the mechanics of this multi-kingdom ecosystem provides the precision required to intercept pathogenesis directly at its source. Dysbiosis functions as a compounding, destructive feedback loop between the gut and host organs. As the diseased liver loses its ability to send regulatory bile acids into the gut, severe ecological collapse occurs. Rogue oral microbes, like Streptococcus, colonize the lower gastrointestinal tract, pumping accelerated volumes of toxic metabolites back to the failing liver, completing the toxic loop. Pharmacological strategies must treat the gut microbiome as the primary target for intercepting systemic disease. Mastering the mechanics of this multi-kingdom ecosystem provides the precision required to intercept pathogenesis directly at its source.